All right, today we're going to talk about Chapter 12, the non-living pathogens, viruses, viroids, and prions. Not all pathogens are cellular. Many infections of humans, animals, plants, and even bacteria are caused by acellular or non-cellular particles. These are non-living pathogens. Acellular infectious particles include viruses, viroids, and prions. What is a virus? Well, a virus is much smaller than a bacteria. It's usually measured in nanometers as opposed to microns. Acellular infectious agents have either DNA or RNA, but they don't have both. They cause many infections in humans, animals, plants, bacteria, and they cause many of the diseases that affect the industrialized world. Some examples of viral disease are the common cold, influenza, herpes, SARS, polio, HIV, and coronavirus. All right, so here's some relative size comparisons of selected virons. So here's a mammalian red blood cell. They're about 10,000 nanometers in diameter. Here's an E. coli bacteria. E. coli is about 1,000 by 3,000 nanometers. And then next to that, you can see a smallpox virus, a bacteriophage, tobacco mosaic virus, and these teeny tiny guys down here are poliovirus and bacteriophage MS2. Very, very small. Some of these smaller ones are even on par with the size of bacterial ribosomes. Viruses cannot carry out any metabolic pathway outside of a host cell. They can neither grow or respond to the environment, and they can't reproduce independently. They recruit the cell's metabolic pathways to reproduce. They have extracellular and an intracellular state. So the extracellular state, or the naked state of a virus, it has a protein coat or a capsid surrounding its genetic material. It can also have an enveloped state. So it has a phospholipid envelope which surrounds the capsid. The outermost layer provides protection and recognition sites for host cells. In an intracellular state, the capsid are removed and the virus exists inside the cell only as nucleic acids. So either DNA or RNA. So here's a model of a naked virus anatomy. Here's a capsid. It's sectioned so we can see the inside. These little balls are called capsomeres. They're the subunits that make up the capsid. And inside that is the nucleic acid, the viral genome. And it can be either DNA or RNA. An enveloped virus looks similar on the inside. Here's our capsid made up of capsomeres with nucleic acid in the center. But this enveloped virus has an envelope that surrounds it. And this envelope may even have spikes that help to recognize host cells. So how do we differentiate viruses? Well, we can differentiate them from one another based on their genetic material. The viral genome may be DNA or RNA, but never both. So there can be double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. Their DNA can be linear and segmented, or single and circular. For example, the influenza virus genome has 8 linear segments of single-stranded RNA. Cells always have double-stranded DNA, eukaryotic cells, and bacterial cells. Single-stranded DNA and double-stranded DNA double-stranded RNA, are almost non-existent in bacterial or eukaryotic cells. All right, another way to differentiate viruses are by their hosts. Most viruses infect only particular host cells. They're species specific. For example, dog viruses don't infect humans. They may be so specific that they infect only a particular kind of cell within that specific host. For example, HIV attacks helper T lymphocytes in humans, but doesn't infect muscle or bone cells. There are also generalists. These are viruses that infect many kinds of cells in many different hosts. Things like rabies. All right, so here's some other examples. We see tobacco mosaic virus in an infected leaf on the left compared with a healthy leaf on the right. Bacteria can also be infected. So the bacteria in this image is this blue-gray shape here, and it's under attack by multiple pinkish bacteriophages. And down here you can see a picture of human white blood cells, cytoplasmic membrane, with HIV particles in blue attached. So host specificity is due to viral surface proteins, which have a precise affinity or attraction for complementary proteins on the host cell membranes. A lock and key theory, not unlike enzymes and their substrates. There's a video I'll link for you guys on D2L to watch about differentiating viruses based on host specificity. 3 mechanisms of entry of animal viruses overview. So let's look at this. There can be direct penetration, where the virus has receptors. The virus attaches to these receptors on the cytoplasmic membrane, and the genetic material of the virus is brought inside the cell. So here's the viral genome. The capsid's left out here on the cell membrane. There can also be membrane fusion, where the viral glycoproteins match up with receptors on cytoplasmic membrane of the host cell, and the envelope dissolves in the membrane of the host cell, releasing the viral genome into the cell. The viral glycoproteins remain in the cytoplasmic membrane. All right, and then the last one is called endocytosis. We have our enveloped bacteria here. It's matching up with receptors on the host cytoplasmic membrane. And the cytoplasmic membrane of the host engulfs the virus. The whole thing, envelope and all, capsid and genome, make it into the cell. Once inside the cell, the envelope and capsid open up to release the viral genome into the cell. So these are the 3 different mechanisms of viral entry into a host cell. Animal virus replication. So replication of animal viruses is based on recognition and attachment. So the capsid or the envelope proteins recognize the host cell receptor. So this is how the virus finds a host cell. Then there's entry. So there could be fusion with the cell membrane or endocytosis. And then synthesis is the next step. So DNA from the virus enters into the nucleus, or if it's an RNA virus, into the cytoplasm. The host cell assembles new viral particles. And finally, the last step is release of all those newly assembled viral particles back out into the extracellular space to go and infect more host cells. So this release can happen by budding, by exocytosis, or the cell itself may become so jam-packed with viral particles that it bursts and lysis happens. All right, replication of animal viruses. Enveloped viruses They cause persistent infections. They're released from the cell by budding, so they don't destroy their host cells. Naked viruses are released by exocytosis or lysis. So here's an example of budding. We have a newly formed viral capsid inside the cell. It pushes up on the cell membrane and is coated with membrane and viral glycoproteins. And it begins to bud off. It pushes up and up, membrane encloses around it, and eventually it leaves the cell as a fully enveloped viron. Let's talk about bacteriophage replication as an example. Lytic replication. This replication cycle usually results in lysis and death of the host cell. And the basic stages of the lytic replication cycle are recognition and attachment, entry of viral genetic material. The host chromosomes are degraded. There's synthesis of new viral particles. They're assembled and released from the cell, usually by lysis and death of the host cell. The lytic replication. So here's another example with pictures. So we have first attachment on the outside of the cell. The genome of the virus enters the cell. The phage DNA is in the cell and the bacterial chromosome becomes degraded. The host cell produces or synthesizes new viral particles. They're assembled and then they're ready to be released by the cell as they burst through the cell membrane, destroying the host cell. Viral replication also includes lysogeny, or a modified replication cycle, where the infected host cells grow and reproduce normally for generations before they lyse. Inactive bacteriophage is called prophage. And induction occurs when the prophage is excised from the host chromosome. Induction can occur through DNA damaging chemicals, UV light, or X-rays. And after induction, the lytic cycle will occur. So here's an example of the lysogenic cycle. We have attachment, entry of the genome. We're building, the cells producing viral particles, they're being assembled, and they can be lysed. So this is like the lytic cycle, right? But you can also have attachment, entry of the DNA, and both the bacterial cell and the viral genome, they combine, right? So now there's just one chromosome. The bacterial cell is allowed to continue and divide, replicating and spreading the viral DNA throughout daughter cells. Replication of the chromosome and the virus by cell division. And then something happens, some cue that induces the lytic cycle, where it begins to assemble viral particles, synthesize them, assemble them, and then release them all at once. This lysogenic cycle is particularly insidious. All right, so a general idea for transduction is we've got a viral genome that's inserted into a bacterial cell. The viral DNA takes over and the nucleoid, the bacterial DNA breaks down, right? And the bacterial cells begin to assemble and synthesize more viral particles, right? So these viruses can exit the cell, but there's something different here. Instead of having all viral genomes here, this one is carrying bacterial DNA. So by chance, sometimes some of that fragment of bacterial DNA makes it into a virus. So that's transduction, when a virus carries bacterial DNA with it. The role of viruses in cancer. Viruses cause 20 to 25% of human cancers. Specific viruses are known to cause certain human cancers like Kaposi's sarcoma and cervical cancer. All right, are viruses alive? So infectious agents with both living and non-living characteristics, they can reproduce, but only when they're inside a living host cells. They can mutate, but they're not made of cells. They have no cytoplasm or organelles. They can't carry out metabolism on their own, and they have DNA or RNA, but not both, like living things. Other parasitic particles include viroids and prions. Viroids are extremely small, circular pieces of RNA that are infectious and pathogenic in plants. They're similar to RNA viruses, but they lack a capsid. And there's no known animal diseases caused by viroids. So here's one effect of viroids on plants. You can see a major difference in the size and texture and structure of these potatoes infected by a viroid. Prions are the third type of infectious particle. Prions are only protein. They cause spongiform encephalopathies. Okay, so prions are only protein. They cause spongiform encephalopathies. Things like mad cow, scabies affects goats and sheep, kuru, and Creutzfeldt-Jakob in humans. It's resistant to proteases, UV light, heat, and disinfectants. Prions are only destroyed by incineration or autoclaving in sodium hydroxide. So these can cause real problems, and they're incredibly difficult to manage. Characteristics of prions are they cause diseases with fatal neurological degeneration and loss of brain matter. Large vacuoles form in the brain, creating that characteristic spongy appearance in brain tissue. Proteinaceous infectious agents, cellular PRP protein made by all mammals. Normal structure has alpha helices called cellular PRP. Alpha helices are these twisty structures in the protein. Those are called an alpha helix. Prion PRP, or disease-causing form, has beta-pleated sheets called prion PRP. And you see here these beta-pleated sheets are folded, kind of like if you fold a piece of paper to make a fan. Prion PRP changes the shape of cellular PRP, so it becomes prion PRP. Alpha helices replaced by beta-pleated sheets. All right, so that wraps up our discussion here of viruses, virons, and prions. Please come prepared with questions to office hours or lab time.